This session is titled Epigenetic Editing, the Next Leap in Precision Medicine to Cure Patients, with our keynote speaker, Amber Salzman. I'm going to introduce our speaker. Okay, here we are. How are you, Amber?
Good morning.
Yes, it's morning for you. It's early, right? And 8:30 for you. Yes. Great to see you here. I don't know. Were you able to catch any of the
Yes, I
competitors?
did. Yeah, it was great to
You
hear
did.
some of the competitors. Wonderful. Always exciting work going on.
Yeah. All right. Great. Well, let's get started with the session. Here's some of my, initial thoughts as I frame up this discussion, this interview with you, which is a huge opportunity. Medicine is very personal. The patient-doctor relationship is transformative. Very often, physicians do not have the solutions for a lot of very challenging patients. What we've done over the course of the last decade or longer is we've actually now enabled a lot of newer treatments for very difficult to treat patients. And as a result of researching rare diseases and rare conditions, we've made tremendous advances in medicine as a whole, in learning about others as well. So it's my pleasure to introduce Amber Salzman, who is a PhD, is CEO of Epicrispr Biotechnologies. She's a leader with more than 30 years of experience in the pharma industry. She served as the president and CEO of Ohana Biosciences. And prior to that, she was also president and CEO of Adverum Biotechnologies, was a co-founder of Annapurna, where she served as president and CEO before its merger with Avalanche Biotechnologies to become Adverum. During that time, she saw the company's growth double. She began her career at GSK in research and development, where she was responsible for operations and drug development across multiple therapeutic areas, overseeing a very large pipeline of drug development. Trials with over 30,000 enrolled patients, managing 1,600 employees, and a $1.25 billion budget as well. Following her time at GSK, she served as CEO of Cardiokine, a pharmaceutical company that developed treatments for the prevention of cardiovascular disease, and saw the successful sale of the company to Cornerstone Therapeutics. She also currently serves on the Osler Diagnostics and AviadoBio UK boards. Her background is in mathematics. She holds a PhD in mathematics from Bryn Mawr College and also received her bachelor's degree from Temple. In addition to advocating for patients living with rare diseases, she leads the Stop ALD Foundation, a nonprofit medical research foundation that's focused on developing novel gene therapies for adrenoleukodystrophy, ALD. She's making tremendous waves in redefining precision medicine, going from cutting DNA to actually switching genes on and off. And so, Amber, once again, great to have you. I want to dive right into our interview and ask you about your transition. You started in R&D, right? And you then became a commercial leader, responsible for the business. Can you share some of those moments that became life lessons?
Thank you, first of all, for having me. Wonderful to be here. And I will say, I feel like I grew up at GSK and was really fortunate to work with wonderful people who I learned a lot from them. And there were certain moments I feel that shaped me. I mean, one moment that I think of when I was on the R&D executive team, the chairman of R&D, Tachi Yamada, who I was very fortunate to have as a mentor, he came in one time and he was reading a letter. This was post 9/11. And we had all actively worked to get Adverum on the market for asthma and COPD. And it was, we were in tears. He read a letter from a patient who said that if it wasn't for Advair, he would not have made it out of the towers. That's how significant this kind of medicine could be. And while it seems like just one moment in time, it was those kind of moments that it was like, I don't care what we've got to push harder, because there's patients that are completely dependent on people like us. And if there's anything we could be doing, we should be doing it. And then fast forward, not that much later, I found my family was affected by adrenoleukodystrophy. And I had worked on very much more common disorders, diabetes, asthma, CBD, much more common, depression. And then to all of a sudden be struck with a rare neurodegenerative disease, I just got thrown into a world that I, to be honest, wasn't even familiar with, and realized there wasn't enough work being done. And when I knew that it was too late for my nephew and I was going to lose him, and we tested other family members and saw the bomb was going to go off and 2 more were going to die, I thought I had to dive into doing more research in that area. So I was very fortunate to be surrounded by people that helped. And my day job, I still had to lead development GSK. My night job was doing everything I could to drive development for drugs for rare diseases, and in this case, affecting my family. And it was, I guess, when you ask the transition to biotech, I guess I would say I owe a lot to Tashi Amada. He was the one that always thinks 10 years ahead of everyone else. And I remember how you have your career development annual review with your boss, and he's like, Amber, your next job should be CEO of a biotech. And I was like, what? And he was right. And he really helped direct me to be able to have an impact and really push things forwards very rapidly.
Well, that's the power of mentorship. Having you believe in something that you didn't even imagine or didn't even consider pursuing, right?
Yep.
Yeah. So we met at Financial Times, where I heard you speak, and there was something powerful behind those words. You strike me as a person who is a leader with a purpose, with urgency and actionability. What is your particular approach to making decisions? And am I right in?
Yeah, I mean, look, I'm really fortunate that even right now at Epicrispr, my husband's family is affected by facioscapulohumeral muscular dystrophy, and I hadn't been able to do anything for that. I'm now fortunate to be in a company where, when I joined, a little more than 3.5 years ago, the team said we could address FSHD, and in 3.5 years, we're dosing patients and helping patients. That doesn't come without a sense of urgency in caring for patients. So what I've learned over the years is you've got to surround yourself with the right people. And I am so privileged to have an unbelievable team. Because, I mean, you mentioned that I have a PhD in math, which usually I'm embarrassed to say, but since you mentioned it, one of the things about math, when you do a problem today, tomorrow, next year, the solution is the same. You're going to get the same number to the decimal points. Human biology is not like that. There's a lot of variability. So even if you do the same bench experiment today and do the exact same thing tomorrow, the results will be hopefully very similar, but they're not identical to the nth degree. And if we just take that as a basis, patients, there's a lot of variability. And it takes the right kind of people to be able to look at a slew of data and figure out what story that data is telling you, and then go back and verify that you got the right story. So that's one, really surrounding yourself with people with that kind of experience and vision and stamina. And then also having the people to say, look, we're never going to have all the answers, and we can't be irresponsible. These are human lives, but they're also waiting on us. So when do we know enough to know that it has a really good reason to be of benefit, and have a really good reason to believe that the risk out is nothing compared to the benefit? And that's really, as I've gotten older, I must say, I used to think, 50-50, you have to have a good science, good technology, 50% good people. And I keep changing that. I feel like you have to have 90% of it is the people and 10% is you start with the right thing. And I don't mean to minimize. Look, we leverage the work that Jennifer Doudna, obviously got a Nobel Prize, and others did. So I'm leveraging other people's brilliance in terms of the technology. But then you've got to surround yourself with the people who can really be focused. And I guess in my case, I surround myself with people who are so driven to help patients and do it with that sense of urgency and mindset that they're waiting on us. And how do you responsibly develop medicines for them?
Yeah. Yeah, I heard James Madera, the CEO of the American Medical Association, just recently at the New York Bio Innovation Summit, and your bio is partnering with Amadev Pharma and with our board on this event, talk about that difference that you just described as well, that humans, we're biological systems, yet we want to make decisions as if we were engineering systems, which we're not, right? So that triggered kind of that kind of perspective as well. Since you alluded to the technology a little bit, and you're making just tremendous leaps. All right. What is epigenetic editing, and how is it different from CRISPR or other precision modalities?
Well, first, I will say that there is a little bit ageism. People that went to med school and PhDs a while ago, we really didn't talk about the epigenome. We talked about DNA and genes. And you talk to younger people, even non-science majors, are like, of course I know about the epigenome. It's above the DNA. So just to add to that context for the different education, I mean, look, I have the same DNA I had when I was 18, but I don't look the same as when I was 18. And that has to do with environment and epigenetics. So when you look at it from that perspective, if you can control the epigenome, which is really how your DNA behaves, so how the genes express or don't express, if you can control that, so you can regulate the gene expression or any genes anytime, that's a real superpower. Of course, one of the huge limitations is we don't understand enough of what's going on. But in the cases where you do, and you do understand where the epigenetics are making your gene expression either too low or too high, then we can have a huge control. So when I mentioned I use CRISPR, so what CRISPR is really an unbelievable system. Look, I did small molecule development for many, many years. It took a long time. And you tried to be on target as much as possible and as off target as little as possible. But you really weren't working at that kind of precision. With the CRISPR technology, you can very rapidly program your CRISPR-Cas molecule to bind to the precise location you want to correct. So that's sort of the traditional CRISPR-Cas technology. What we did, because it's based on the work of our scientific Stanley Chi, who worked with Jennifer Doudna on the CRISPR patents, he said, okay, well, if we get to that precise location, is there something else we can do besides cut DNA or edit DNA? And his approach was, let's not risk genomic instability. Let's deactivate that Cas molecule nuclease. And instead of cutting, let's fuse a gene modulator to it and control what the genes do. So it adds this level of precision where it's not just on and off. You can almost tune how the gene expresses, and you can do so in a relatively safe way because you're not disrupting the genome. You're really just modifying how the genes express.
Okay. How was the journey to discovering that technology? Did you encounter any challenges?
Yes, huge challenges. So as you know, you have a technology, but then in this case, for example, so I talk about this CRISPR-Cas molecule and being able to bind to the right location, but how is it delivered? And if you think about our lead indication, which is a muscular dystrophy, we've got to get that machinery to your muscle cells. Well, with today's technology, the best way to get to the muscle cells is with an AV vector. As we've seen, there's products that have been approved that do just that. The challenge with that AV vector, it's got a really small payload, and the Cas molecule and its machinery is fairly large. The other challenge is the Cas9 molecule is bacterial derived, so it could cause an immune response. So what we were really fortunate is we licensed a kind of next gen, as I would call it, Cas molecule from our scientific founders lab at Stanford, Cas Mini. And the big advantage of Cas Mini is that it's really tiny in comparison to Cas9, and it's also non-bacterial derived. Our team then started with Cas Mini and did further engineering, so it was even smaller, more active, but still did not have that bacterial origin. And when we looked at it in comparison to, for example, Cas9, it didn't have that kind of immune response. It was really clean because it was non-bacterial derived. So that was just one of the challenges. Then we needed to find the gene modulator. In the case of FSHD, what happens is there's an area that's hypomethylated, and it lets this poisonous DUX4 leak out and kill your muscle cells. So we needed to methylate it and stop DUX4 from leaking out and killing muscle cells. Well, so we figured out with the CRISPR-Cas molecule, in this case, a variant of Cas Mini, how to program it to go to the right location. But then we had to fuse a molecule to it that could, in fact, suppress that DUX4 expression. So we did development in that area. I would say then once we had something that looked like it worked, what's really challenging, going back to the human element, I mean, patient or medicine, patients are so different. So there's so much variability. So if you want to see that it works, you know, in some patients, it really affects their upper body much sooner and more severely. In some patients, it's actually the lower body. You know, in some patients, it's facial. They can't smile. They can't close their eyes. So if you are actually having a positive effect, how do you measure that in a way that you can show that your drug is working? And as you know, we always try to pick a good endpoint, but the same endpoint for one patient may not be the right endpoint for another patient. So that presents challenges, especially in rare diseases where you're not going to enroll hundreds and hundreds of patients. So, but I will say this is where there's advanced technologies. We're very fortunate. I mean, we're looking at a lot of different endpoints, and we learn from other rare diseases where they do things like win ratio, where if you hit this or that, depending on the patient. But the other thing we did is we partnered with Springbuck Analytics, and they figured out a way to do whole body imaging and look at your muscle fat fraction. So overall, if your muscle is turning to fat somewhere, but the measure that you pick to look at doesn't capture it, you're still going to see it. So they were able to do this whole body imaging analytics, feed in a whole bunch of natural history of FSHD patients. So you could be predictive for any of our patients that we treat at baseline. If we didn't have benefit, what would their fat fraction look like if you keep going? But again, that's really leveraging. I mean, there's quite a bit of work to do to improve upon their machine learning and make sure that the FDA sees the correlation between the fat fraction and a functional measure that's important to patients. Because, I mean, imaging only matters if it helps you function. So, you know, you really have to constantly be looking out for better technologies and better approaches to appreciate how to get something to help patients, even though they all have their particular uniqueness, which, of course, we love our uniqueness, but it's also a challenge.
Yeah, and it's exciting to see that you know, the treatment is now in human clinical trials already, right? Recently.
Oh yeah, we're juicing.
Yeah. So I probably would be remiss if I wouldn't ask you on your opinion about Sarepta. I've spoken to some of our expert judges who are also on the board, or scientists, John Hemming, who had brought this up as well. Can you make sense and provide your perspective of some of the disappointment?
So look, I always feel like we have to learn and not walk away from challenges, and it's tragic that there's been a loss of life. Tragic. But we can't then say, walk away, this is dangerous. Let me just start with the premise. I mean, look, no one's going to tell you chemotherapy is safe, but if you need it, you'll take it. And again, no medicine, I mean, that's kind of a big example, but there's risks for everything. So look, in the case of Sarepta with Elevidys, Duchenne muscular dystrophy is it's a bad disease. So you do, even though we know that AV is dangerous, you do want to risk it. However, when I look at the 3 deaths that did occur, 2 in Duchenne's and 1 with their limb-girdle trial, there's a couple of things that come to mind. One is that these patients were very advanced, so they had a lot of muscle loss. So when you don't have muscle, it turns to fat, and fat is not vascularized either. If you're dosing based on body weight rather than muscle mass, so you're taking an advanced patient, an adult who weighs a lot, and you're dosing based on that, you're flooding them with a huge amount of vector. And where's the virus going to go if there's no muscle to go to? It's going to go to the liver. So that's a big challenge. So I think that was one thing they got caught up in. I think the other challenge, and this is, I'm just going by publicly disclosed information, is that as part of their manufacturing process, and people struggle with this. We're very fortunate with our process. People struggle with the fact that when you try to put the gene of interest inside those AAV capsids, some of them end up not making it in and you have empty capsids. If you have too many empty capsids, then you're literally having to dose people. Let's say half your capsids are empty. You have to give them twice as many capsids just to give them the dose you want. And from what I understand from, again, publicly, you know, FDA has talked about it when they got accelerated approval, they did have a lot of empty capsids. So you're giving them a lot of viral load, and you're dosing them based on body weight, not based on muscle mass. There's risks. There's risks involved. I would say the third thing, which really gives me some comfort based on us, using AV to get to the muscle, is that in Duchenne's patients, unfortunately, when you want to check that somebody's liver is healthy, you look at ALT and AST levels. Unfortunately, for Duchenne patients, at baseline, they're not normal because of their muscle degeneration. So for the most part, it probably is the muscle and not the liver. There are other ways to look at the liver, but it could potentially be masking a liver problem. Where with FSHD patients, they do have normal ALT and ASTs in baseline. So if we do see elevation of baseline, they're probably not a candidate for this kind of treatment. So just to add to that, look, Sarepta has dosed on 1,000 patients. And the patients they lost are tragic, but there are a lot of families that are incredibly, incredibly grateful for what it has done for their kids. Now, I think we should be able to learn from it and do better. These kids are still progressing as rapidly. There's still a huge unmet need, and we need to do more. But I would hate to throw the baby out with the bathwater. Like, take the challenges, take these tragic events and learn from them, and try to figure out who are the right patients and how you can change things so that you can dose responsibly and address more. So I think my only call here is that not just around all companies, I mean, there's been a lot of tragedies. Unfortunately, there's been a lot of miracles too, is that we share information and are transparent so we can all work on this together and figure out how to fix it as opposed to run away when there's patients depending on us. I know I probably answered that too long, but I do say, look, AAV, anybody that tells you that AAV isn't risky is not being transparent. It is risky. So I say use it responsibly and be transparent about it, because some of these diseases warrant that kind of risk.
Yeah, absolutely. So I started our discussion with talking about the fact that medicine is personal, and it's quite personal for you as well. Can you describe some of your personal experiences with patients with FSHD to the extent that you would like, and tell us how that impacted your personal journey as a leader?
Yeah, well, as you know, I got into genetic medicine because I lost my nephew, and at the time that he was diagnosed, genetic diseases, it's a family party. We all get diagnosed. And then unfortunately, my son, who was 1 at the time, and another nephew tested positive. So it was really personal to try to come up with a treatment in time for them not to suffer what my nephew suffered from. So as I said, I very much focused on, and through a public and private partnership, we ended up progressing the gene therapy for ALD, Skysona, which got approved. Now that took 20 years. That's way too long. My son and my other nephew ended up having to have a stem cell transplant, which is also very, very dangerous. And there's long-term ramifications as a result. So 20 years is not, that's just not going to cut it. So I've tried to figure out how to work more rapidly. When it came to FSHD, as it happens, my husband's family suffers from it. And initially, when I saw the symptoms, they, as you know, with a lot of these diseases, it takes a while to be diagnosed. So initially, they didn't have the right diagnosis. Then they finally did get diagnosed with FSHD. And I saw first his father's sister, who gait disturbances that progressed. The kids had symptoms that progressed, wheelchair, etc. Next generation. And that family said to me, look, you worked on ALD. Why can't you work on FSHD? And at the time, the challenge was the root cause wasn't understood. The genetics and epigenetics of FSHD weren't understood. And great help from others. About 15 years ago, some seminal papers came out where it really the understanding of FSHD was validated. And as my cousin-in-law says, I had literal skin in the game for them to figure that out, because they took biopsies, etc. And when there was a target, then I could go after it. I'm not a bench scientist. I'm a drug developer. And as soon as there was a target and I saw the kind of technology that Epicrispr had, it was like, we can fix this. And it is just, I mean, while it's scary to be dosing patients because this drug has not been in humans, and my huge admiration goes out to these patients. I'm so grateful for them. And look, we won't know if it works. It takes time. That's why we do clinical trials. But I'm so optimistic and so grateful for everyone that's been involved to move this field forward.
Yeah. And your son graduated, and right? And
he actually graduated, and he works as a behavioral therapist in an autistic clinic. Interestingly, we just went to a cord blood reunion at Duke, and there were a bunch of adults who got transplanted as kids from various diseases, and every single one of them worked in a field that was helping people. So I mean, it could have just been coincidence. The numbers weren't there, but it was interesting that these adults, I say kids because they were transplanted as kids, these survivors all were having such an impact on other people's lives. It was very interesting to hear what they were all doing.
Yeah. So how far away are we from seeing patients with FSHD now having access to your treatment?
Yeah, well, we just started dosing. We'll have very early clinical results early next year. The full first-in-human trial concludes in the beginning of 2027. You never know until you see how strong the signal is, what it will take in terms of how many patients it would take to actually show efficacy. And as we talked about, there's variability. But if we were fortunate in terms of signal, and it's always, sometimes you just enroll very variable patients, sometimes you don't, and you can't control. It could be potential market in the 2029 timeframe, but in the meantime, we're making it accessible. People are signing up for the clinical trial. I will add, one of my big frustrations is right now, because we want to reduce variability, it's only open to ambulating patients. And it's also only adults, and that has to do with, you have to show reasonable safety in adults before you go to kids. I do want to find a way as quickly and as responsibly as possible to move into the broader population, because kids really need it. And also non-ambulating patients don't want to get worse.
Okay. And it's a small population of about 45,000 patients in the United States.
In the US, they say there's 45,000. As we have found with other rare diseases, when there's no treatment, it's rather undiagnosed, but at least those are kind of the numbers that are being bantered about now. But the thought is that there's more.
What other kinds of conditions is your drug development potentially considering in terms of other indications?
Yeah, because we sort of have the superpower where we can upregulate or downregulate gene expression, and we can do it with ultra compact constructs. That opens up the door to some indications that otherwise couldn't be pursued. So, for example, we're looking at retinitis pigmentosa 4, which is a terrible eye disease that progresses. And there's a lot of different mutations, but because we're just changing the transcription, we can be mutation agnostic. So we can go after all the mutations with one product and stop the bad protein from expressing, and still have enough room to deliver the healthy gene, and do that all within a single AAV delivered to the eye. I would say the other area I'm super excited about is where it turns out, and I will admit, I learned this all fairly recently. I have a wonderful team. There's a bunch of really bad diseases where when you have a mutated protein, your body actually has a compensatory gene that if that could be overexpressed, it would make up for the damaged one. Now, the one that most people probably know about is, look, dystrophin is too big to put in with an AAV, and that's why people are putting in microdystrophin and mini dystrophin, which still isn't dystrophin, and that may be why the kids get better, but not completely stop the disease. But there is a gene, utrophin, which if you overexpress utrophin, it would compensate for the lack of dystrophin. It's also big, so you can't put it in. But the fact that we have the power to upregulate it, there's a bunch, and it turns out there's a whole slew of diseases where you can do that. You can upregulate the compensatory gene to make up for that disrupted gene, and these are genes that are too big to replace. So we have a few indications that are doing just that.
Okay. Well, it's been a pleasure having you as a guest speaker. We invite you to stay, at our Medical Innovation Olympics. There's quite a few panels coming up. I'd like to leave the last words to you, and as you are speaking to many leaders at various different conferences and through your work at Epicrispr, including at our event with our audience. What is your number one focus now? What is one message that you'd like us to, to
Well,
leave us
just for all of us, I think if we stay focused on patients and work with patients, that we
with?
can learn so much and co-develop with them. Look, we're all gonna feel like we hit brick walls way too often, but it takes it takes a lot of perseverance and keeping the patient in mind. And together we can do really amazing things. And I mean, I listened to some talks earlier. I really admire what everyone's doing. Keep up the amazing work, collaborate, learn from others. And I think it takes way more than a village to bring the kind of medicines that we need.